So it's my pleasure to introduce Matt Reid today. It's for Cornell University. And Matt and I first met when I think I was home visiting my parents. And I popped down to Princeton for a day to visit some faculty there and met with Peter Jaffe's research group, and Matt was a PhD student at the time. So probably 10 years-ish? Over 10 years.
A little over 10,
yeah. So Matt did his undergraduate degree at the University of Chicago in chemistry. Did his PhD, well, then he got into Peace Corps for a couple of years.
Yes.
Tanzania?
Yes.
Correct, yeah. Did his PhD at Princeton. Paige and I tried to hire him as postdoc. He decided to go to Switzerland and go to EPFL. Can't blame him for that. And then he's been at Cornell for about 10 years now. No, not, yeah. 7 years now.
Yeah.
And his work is in biogeochemistry, water quality. And he's going to be talking about nitrogen and non-point pollutants today. And Matt is one of our NRT circularity speakers this semester, so he's going to touch on that a little bit. And our theme is mining, and you might look at this and go, there's no mining here, but nitrogen is a resource we mine a lot. It's just out of the atmosphere rather than out of the ground. So. And the last quick point, if you are here if you're part of the NRT cohort and you need to sign in for a seminar, sign-up sheet is in the back left corner there, so you can get meet your quota. So with that, I'll turn it over to Matt. Welcome.
Thanks so much, Bill, for the nice introduction. I remember when you were in Princeton, I remember your talk. Princeton at that time was all about like ecohydrology. I felt like I was the only chemist, and Bill came and gave a talk, and there were pKa's and chemical structures, and I was like, okay, I see a role for myself in the environmental engineering community, because Princeton at that point was not so inclusive of chemists. So thanks everyone for being here. It's really my pleasure to be here to talk to you today about what my group has been doing in the past couple years on thinking about creative new ways to manage non-point source nutrients in agricultural systems. Okay, so before I start, I just really want to acknowledge the people who actually did the work that I'll be talking about today, a group of graduate students and a great group of undergraduate students who have also contributed. And my talk today will strongly feature the work of my student Zihao, who's actually here, because one of the reasons we're here in Minneapolis, in Minnesota, actually, is as part of a USDA project. So he came with me to work with some folks over in St. Paul to get some field projects off the ground for next year. So my group at Cornell works on biogeochemistry and ecosystem engineering. And from this sort of figure, I think some of the things I want to try to emphasize is that one of the features of our group is that we work across scales. Okay, so we do work at the molecular level. We also try to bridge that to like field scale observation and monitoring. We combine modeling with experimentation. And in a lot of talks, this is when I would say, okay, now I'm going to talk about either nitrogen, which is one research theme in my group, or I'm going to talk about arsenic in rice paddies, which is a different research theme in my group. And actually, in the talk today, I'm going to integrate these in a way which is actually motivated by the fact that this is a seminar on circularity, which I don't usually talk about, but I've made an effort to incorporate more aspects of circularity. And so I'm excited, actually, in this talk that I'll be able to couple these two different aspects of my research program. Okay, so I think in this audience in environmental engineering, I don't need to spend too much time motivating why nitrogen matters, why non-point source nitrogen is particularly difficult to treat due to its diffuse nature, or to talk about some of the costly environmental impacts that result when reactive nitrogen is released from agricultural and urban systems into water. This is not a new problem. Okay, we've known about issues related to eutrophication and nutrients for a long time, but it still represents a major unmet treatment need, especially where we are now. Okay, in the Mississippi Basin. Okay, there's a huge unmet treatment need for reducing non-point source nitrogen from agricultural watersheds to address hypoxia in the Gulf of Mexico. In the east, Chesapeake Bay TMDL is a similar situation. And despite significant investments in best agricultural, best management practices, edge of field practices, and this is a little bit old, but still to this day, there's not really a meaningful reduction in the amount of nitrogen that's making it to the Gulf of Mexico. So one of the culprits here is agricultural tile drainage. So agricultural tile drainage is really important because it allows farmers to control the water levels in their fields, okay, so they can drain the fields in the spring, plant when they need to plant. But one thing that happens is that when there is precipitation or snow melt, that water is sort of shunted quickly to streams, and it bypasses a lot of the natural filtration functions of soils or riparian areas. So it kind of like short circuits a lot of those natural processes and transport the nitrogen directly to streams. One really important thing I want to emphasize is that agricultural drainage water is really different from municipal wastewater, okay? The carbon to nitrogen ratio is very low. The nitrogen is primarily in the form of nitrate. We don't have a lot of ammonium, okay? So we need to think about managing this differently than we may think about based on what many of us have as more traditional training in sort of like municipal wastewater treatment, where we have much more carbon, all right? And a lot of the story today is going to be about carbon and where we get that carbon from. Okay, so tile drainage infrastructure is widespread across the US, especially in the upper Mississippi Basin, but also in the Finger Lakes region. You can see over there where I live, there's also quite a bit of tile drained agricultural land there. And nature-based systems, many of you have probably heard of nature-based systems, things like constructed wetlands. I'll be talking about these denitrifying bioreactors are suitable for this type of source because they're passive systems, relatively low cost, that essentially utilize naturally occurring processes to address some of these water quality issues. So these are sometimes called nature-based solutions. I don't really like solutions, so I call them nature-based systems, okay, but like NBS, okay. Prominent examples are constructed wetlands, okay, are a really popular form of nature-based system for managing nitrate. This is a picture I took a couple years ago in California, this like living levee concept. These are being built on San Francisco Bay. And so for nitrate, if we want to stimulate denitrification, the key things that we need like saturated conditions, okay, so that the water is hypoxic to anoxic, okay, so that we can have microbial denitrification. And the other thing is we need a source of carbon. These are sort of like the two pieces of the recipe that we need to stimulate effective denitrification. So constructed wetlands are good for that because you have flooded land, decomposing plant material that provides a carbon source. So another way of doing that is increasingly popular in agricultural systems is an edge-of-field practice called denitrifying woodchip bioreactors. So this is basically a trench that gets built, that gets dug out, filled with often wood chips, but it could be any type of lignocellulosic biomass residue. Okay, so this is kind of where we're bringing in the first aspect of circularity here. Okay, how do we make beneficial reuse of lignocellulosic waste material? Okay, one way is as a carbon source to stimulate denitrification. Okay, and the way the systems work is so the water comes in here, gets routed through this wood chip bed. There's a water control structure at the outlet that usually maintains the water level at a high enough level so that the beds remain flooded. Okay, so these are relatively inexpensive to build compared to many types of engineered systems using lignocellulosic biomass residues. And then the idea is that the decomposition of that biomass provides a source of labile, bioavailable organic carbon that can then fuel the denitrification process, okay, the sort of sequential reduction of nitrate through a series of intermediates, including nitrous oxide, which I'm interested in, but I'm probably not going to talk too much about nitrous oxide today. And ideally, we push this all the way to nitrogen, and having more carbon actually helps to push this all the way to dinitrogen gas, okay? But one of the key questions now is like, okay, lignocellulosic carbon, labile carbon, how do we get from lignocellulosic carbon to bioavailable carbon? And that will be a sort of major topic of what I'll talk about today. So I'll start by talking about some challenges for woodchip bioreactors. Then I'll talk about how we can translate some of our more detailed biogeochemical insights into how these systems are working at like a more mechanistic or process level to thinking about how we can control the systems. And then finally, at the end, I'll bring in rice paddies. And that link may not be really clear to you right now, but hopefully at the end, you'll see why rice paddies relate to nitrate and tile drainage. OK, so I'll start by talking about one challenge, which is that because these systems are passive and the environment is very dynamic, there are challenges related to very dynamic environmental conditions with a static treatment system. OK, so this is work that a really excellent former undergrad, Jenna Israel, did. This was actually kind of like a COVID pivot from her senior thesis to think about how we can model the treatment performance of bioreactors under hydrological variability or seasonal variability. This was done in collaboration with Scott Steinschneider, who's a hydrologist at Cornell. And so we used this field bioreactor. This is about a 20-minute drive from Cornell campus. The bioreactor is underground here. It intercepts tile drainage from these fields. And we had a series of longitudinal pore water sampling ports. And what we basically did is we were able to take samples at different distances in the reactor, translate those distances into travel times using the mean HRT, and then use that to determine a zero-order kinetic for the denitrification. Then we were able to get like a field-based temperature response model for understanding how denitrification rates vary with temperature. This was really important because a lot of times people estimate these model parameters from lab experiments. We were able to model that using field observations. We combined this biokinetic model within a hydrological model where Scott built a machine learning-based model that could translate precipitation into bioreactor discharge, which was actually really hard, because you'll see sometimes there are these like storm events that translate into a large response at the reactor. Sometimes there are storm events that do not translate into a significant hydrological response at the bioreactor. So it was pretty hard to actually develop this precipitation discharge relationship, but Scott was able to do that. And then we combined these into a sort of coupled hydrologic biokinetic model for modeling as a function of water temperature. And this represents the cumulative nitrogen entering the reactor. This is the cumulative nitrogen mass leaving the reactor. So we can model the cumulative load reductions. And then what we did is we combined this biokinetic model with a stochastic weather generator to think about, OK, we have maybe a few years of data, but because this is a stochastic system that depends on rainfall variability, what's going to happen if we look at like 100 years or 1,000 years and try to look at like the statistical properties of how this bioreactor functions? So we did 1,000 simulations. And so that allowed us to sort of compare and examine, we can have like pretty bad years for the reactor where the reactor doesn't work very well. And we also have good years where the reactor is working better, right? Still releasing a lot of nitrogen, but is working better. And some of the key features here to focus on, so these blue bars are showing the daily removal efficiency. So a couple of things you can see is that in the spring, the daily removal efficiency is low, largely because of low temperature. As we move into the summer, we get improved daily removal efficiency, sometimes up to 100%, okay? But when we have storm events here, right, we see that removal efficiency really take a hit, and that's because of shorter residence times in the reactor. Okay, so the water isn't spending enough time in the reactor to get efficient denitrification. So if we have a year like this, where we have sort of moderate flows in the summer, when the temperatures are warm, we can actually get a better removal from the system. Here, right, basically having a dry summer no flow condition, basically the reactor isn't working for us when it can function the best. Okay, so that's one reason why we see relatively poor efficiencies then. And what we could do is we could use this to ask a question about effects of climate change on bioreactor performance. Okay, we know that in the future with warmer temperatures, we expect intensified precipitation. That means shorter HRTs. That's going to decrease the performance of the reactors. At the same time, it's going to be warmer with faster microbial kinetics. So what's the trade-off here? What's the balance going to be? So what we did is we perturbed the gamma distribution for precipitation intensity according to the Clausius-Clapeyron equation for sort of like the increase in precipitation intensity as a function of temperature. We also looked at some local predictions for how mean annual precipitation would change over central New York, and we looked at the baseline range of estimates. The yellow stars represent our 3 years of observations. Okay, so our observations are lining up with these sort of simulation predictions relatively well. And then we can look at a function of different temperatures. What is the effect of, and the thing we can do is we can pull apart using the model these different drivers. So we can look at precipitation intensity by itself, separate from the effect of warmer temperatures. Look at warmer temperatures separately from the effect of precipitation intensity. And so basically what you're seeing is that some of these hydrological changes, in some cases there's a small dip in the load reduction, but this is not a statistically significant difference. Where we see the statistically significant difference is actually when we increase the temperature and looking at those effects of higher temperature. So if you're looking for a silver lining for climate change, it's that bioreactors might work a little bit better because it's warmer and the bugs are a little bit more active. Okay, and the point of, I think, of this first example was to really show where there's a need for improved performance in the reactors, and this is sort of in spring and fall when temperatures are low, and then when storms lead to short hydraulic retention times. Okay, is where we have a need for improved treatment. Okay, another challenge for reactors is that woody biomass breakdown is actually really slow in anaerobic conditions. So this is kind of like a really simplified version. Okay, we have wood, organic carbon, fuels denitrification, but in reality, lignocellulosic carbon is a complex carbon substrate. It has lignin. The lignin really protects the cellulose and the hemicellulose from degradation. Okay, fungi are important drivers of wood decay in the environment. Okay, I'm really happy in my visit today. I had a chance to talk with Jonathan. Jonathan and I had some really great discussions a couple years ago. I learned a lot from Jonathan over the past couple years, so it was great to meet you during this visit. But basically thinking about the need for molecular oxygen to degrade lignin. So we have a little bit of this like this conflict, right? We need anoxic conditions for denitrification, but we need oxygen to actually break down that wood. Okay, so that's one reason why the systems may not work that well. So to explore this a little bit further and to try to understand, okay, how is the wood breaking down? What are the conditions that are leading to, differences in the decomposition of the wood and the role of the wood as an effective carbon source? I worked with some beamline scientists at NSLS2, which is a synchrotron at Brookhaven National Lab, and we put together this. You can think about this as basically like micro XRD. Okay, because cellulose is a crystalline polymer, we can use like XRD with like a micro-focused beam and actually image the crystallinity of cellulose in wood specimens that were collected from different locations in the bioreactor. Okay, so we can compare upstream wood chips from downstream wood chips. Look at how things change as a function of depth. And so the higher the CI, this is the cellulose crystallinity index, a higher CI, so yellow means like non-degraded wood, and the bluer areas are indicating areas of more degraded wood, or at least where the cellulose is depleted. Okay? And one thing you'll see is if you look at this wood chip, we see this really clear area of depleted wood around the surface here as well. But in many of the other samples, we see that there's very little evidence of the wood degradation, actually. And this number here is the average crystallinity index. Okay, so most of these you see the average crystallinity index is pretty high. We've also correlated this mean CI with carbon to nitrogen ratios, which is a more traditional way of understanding the extent of wood degradation, and the mean CI and the C to N ratio are relatively well correlated. Okay, so this is cool. I mean, they're cool pictures, but it also helps us to like really get a detailed understanding of like where the wood chips are decomposing, how they're decomposing around the surface, and also this sense that like it's really these upstream wood chips tend to be showing higher levels of degradation than the downstream wood chips, and this is probably related to the presence of oxygen. Okay, when water is entering the reactor, there's still dissolved oxygen present in the upstream portions of the reactor. Okay, by the downstream, the water is very anoxic. Okay, so right now we sort of understand these spatial patterns as being a result of dissolved oxygen presence in the upstream portions of the reactor. My PhD student Yi is a person who's been leading this, and what we found is that if we then take those wood chips and use batch experiments, different wood chips from different locations of the reactor are supporting very different rates of denitrification. And what we're showing is that these more degraded wood chips have more biomass, have more protein, so more biofilm concentration on the surface, and are also driving faster rates of denitrification. So there's almost like this somewhat paradox or unexpected result that the more degraded wood chips are actually driving faster denitrification, which on the one hand is maybe counterintuitive, but if you think about it a little bit more, it's because they degrade. The fact that they are degrading means that they are releasing carbon, so they're supporting greater biomass concentrations and faster denitrification. And a lot of these other wood chips are really not breaking down, okay? One other feature that we kind of noticed was that sometimes around the pores, there's a little bit of a lower CI, and this may be related to the fact that when the wood chips are subject to fluctuating water tables, the pores of the wood can actually trap air. Okay, so if it's exposed to air and then refloods, the inner pore structure can fill with air, and that could possibly support some aerobic processes from the sort of interior of the wood structure. Okay? So with those sort of now like process level insights, we can think about moving on to the second part. So what do we do about this? Okay, we have maybe more understanding of like what is limiting the performance of the reactor. How can we improve that? So this second part is about some relatively new work my group has been doing on real-time control of bioreactors, okay? So there's sensors, there's control. As a biogeochemist, I kind of see myself as bridging these two sort of more engineering topics. Okay, we can measure things, we can do things like we can pump things into the reactor, we can raise the gates and drain the reactor, but like what should we actually do? Okay, so we have some of these questions where the biogeochemical insight comes in. And the two different control strategies we're going to talk about, one is biostimulation with exogenous carbon dosing, and the second is deliberate draining and reflooding of the wood chip media to enhance bioavailable carbon release. Okay, so biostimulation experiments, we were sort of motivated here by low spring temperatures boosting this performance or possibly helping to improve performance during storm events. We took an approach of thinking about this from like a kinetics perspective, thinking about, okay, like what's the half saturation constant for acetate, and really focusing on actually dosing at a relatively low level. We were concerned about the risk of adding too much acetate, right? Leading to acetate breakthrough, leaving the reactor, we don't want that, possibly leading to biofouling. So we were really focused on just sort of adding relatively low levels to sort of get close to the maximum rates. We had this paired bioreactor system. This is at our field site near Cornell. We injected acetate into this gallery of wells here, and then monitored nitrate in, nitrate out. We had nitrate sensors, pore water samplers for measuring what was happening downstream. This is work led by Tihao in collaboration with Niels Knapp in Cornell's ECE department. So what we saw, so this just shows the temperatures increasing over our 2-month experiment from April 1st, 2023 to June 1st, 2023. The temperatures increased from about 6 Celsius up to about 12 Celsius over that period of time. So it's still pretty cool. Okay, this is important because a lot of times we do. Okay, so yeah, no, Minnesota. So Minnesota, we're down a little bit more, right? But so it's an important point because a lot of times we do experiments in room temperature, right? But this system never gets to room temperature. Okay, we're always at much lower temperatures, especially in the spring when the flows are the highest and the nitrate loads are the highest. Okay, we captured 2 storm events, and basically, this is showing the inlet nitrate concentration. These gray bars are showing where we dosed carbon. The blue is showing the effluent nitrate concentration in an undosed control reactor. And the magenta here is showing the effluent concentrations in the control reactors. You can see very clearly where we dose carbon. We see clear effects of the decreasing the effluent nitrate concentration, okay, stimulating nitrate removal rates, we can look at the ratio of the rates in the experimental reactor compared to a baseline model. You can see we're getting between 2 and like 5 time increase in the rates. Importantly, we tested a period where we dosed at a higher concentration, okay, and we saw no response. That higher concentration was really suggests we were basically saturating the system with acetate, and there was no benefit from adding more acetate, even though from a stoichiometric perspective, you might think we needed more acetate to treat all that nitrate, where we are reaching a kinetic limit there. And then we can compare the cumulative load reductions basically under a baseline scenario, no control, 11.3% nitrate removal, going up to 24% nitrate removal with the control. There were some trade-offs and some risks. A lot of the acetate that we injected was not metabolized and was released in the effluent. This is a problem. It's an environmental problem. It's also a cost problem because the acetate is expensive. Okay, so if we look at the theoretical ratio, the metabolized ratio, this is kind of telling us that some of the acetate was being consumed by something other than denitrification, possibly removing residual oxygen, which was still in the water. But then the total dosed carbon to additional N removed was really high. Okay, so only about half of the acetate that we injected was actually consumed. And when we calculate a cost efficiency, Bill, this is actually because of you. In AESP in Boston, you saw my poster and you were like, how much does it cost? And I was like, that's a good question. So I did a little TEA, because it's a really important point. And so basically, the cost per additional kilogram N removed was about $86, compared to about $33 based on the passive operation. And that compares to a couple bucks, $2 or $3 per kilogram N in a centralized wastewater treatment facility that's using the carbon in the wastewater as the carbon source. Okay, so a lot of this is kind of like emphasizing the expense of the carbon. We could bring this down if we could get to better acetate utilization efficiency, but even if we had 100% acetate utilization efficiency, this is still going to be relatively expensive, which gets us to the next approach, which is, in a lot of ways, it's a much more like elegant approach, which is basically, let's not add anything, let's just control the environment in a way to get the reactor microbial community to do what we want. Okay, provide them periodic exposure to oxygen. That periodic exposure to oxygen can increase the breakdown of carbon, produce more bioavailable carbon, kind of fuel this effort a little bit. There was a paper in 2019 that did this. So DRW means drying, rewetting cycles. SAT means saturated. So you can see that when they do a drying, rewetting cycle, they're able to almost double the nitrate removal rate the day after it's reflooded. And then there's this almost like first order decay over time back to that saturated level. Okay, so we did something similar. We did column experiments where we tried oxygen-oxyg cycling. We also saw about a doubling in the removal rate, but we wanted to really understand why this was happening. So we collected DNA and RNA, sent it to the Joint Genome Institute for sequencing. And what we were able to show here was basically the importance of fungi and the importance of lignin degradation in the breakdown of wood and the release of bioavailable carbon. So what this shows is a time series of different, here we're looking at auxiliary activity, basically lignin degrading enzymes. And so just as a reminder here, this is like the time series. The blue line shows the oxygen concentration. 0 hours is when the reactor was reflooded. So the reactor was drained for 48 hours, was reflooded. We sampled RNA from which it biofilms across this time series, okay, where we do see a decrease in the nitrate removal rate. So we see the highest nitrate removal rate right after the reactor was flooded, which also is a little bit counterintuitive because there's still oxygen in the reactor at that time, right? But that's leading to the fastest nitrate removal rates. So you look at 0 hours, that's when there's still some oxygen left in the system. The highest expression levels of many of these lignin-degrading fungal enzymes is occurring in the 0 hours and 8 hours when we still see some residual oxygen in the system before later times where they decrease to at or below the level that's seen in the continuously saturated reactor. So this is basically the ratio between the expression level in the wetting drying reactor and the ratio in the continuous and the expression level in the continuously saturated reactor. Glycoside hydrolases involved in cellulose decay didn't really show any clear patterns, but when we look at the response of the genes involved in denitrification, or it's also some other nitrogen cycling genes, we see a strong response to the transition from oxic to anoxic conditions, but in sort of the opposite direction as the lignin degrading enzymes. So we see this kind of like complementarity here. Early on oxygen exposure, we see more expression of some of these lignin degrading enzymes, decreasing as a function of time, while we think about some of these key functional genes for denitrification are lowest at 0 hours when we still have oxygen present, but then they increase at later time points, and importantly, they stay elevated for up to 100 hours after the reactors are reflooded. And I think that helps to explain why we did see this sort of like extended period of enhanced nitrate removal, because this relatively short oxygen exposure, we can see, has this sort of like extended impact in stimulating the expression of some of these denitrification genes. And this emphasizes the importance of fungi here. And we have some cool SEM images showing like fungal hyphae colonization of the wood chip structure and breaking down that wood. So then one of the questions here is, how would we operationalize this? How do you implement this, right? Because if we, basically we're saying that if we drain the reactor, expose the reactor to air, we can get better treatment in the future. But when the reactor's drained, it's not giving us any treatment. Okay, so we have now like a temporal trade-off between poor treatment in the future, trading off for improved treatment in the future. How do we do this? Okay, so this was where I worked with my colleague Niels in ECE to develop an optimization framework here. So basically what we can do is we can describe the system. When the system's drained, and then we can look at like the sort of the ratio of the rates, okay? So as the reactor's drained, and these are parameterized using lab experiments, the ratio increases as a function of time. And actually, we did this experiment and then realized we missed the important part of the data set here. We thought we would need to drain it for more periods of time to really maximize that benefit in the rates, okay? But actually, by 48 hours, we're already at sort of like the maximum rate. So we need to go, well, Diha needs to go back and do some more experiments to figure out better like what's happening for these shorter drainage periods. And then when the reactor is flooded, we have this sort of first order decay, which is similar to what was seen in that earlier paper. We could fit the first order dynamics with these two models, and then we basically put these equations into what's called a Markov decision process. So this is basically a system that basically computes all the possible outcomes from decisions at each time point, wetting the reactor, drying the reactor, wetting the reactor, drying the reactor, and basically kind of brute force computes the possible outcomes that we can achieve. And we can run that for a whole series of temperatures and flow rates, since these are the key parameters that control treatment performance, okay? So this is in steady state. And just as an illustration of what this looks like, we can highlight here we're getting a 68% improvement in cumulative nitrate load reductions. Okay, so we are getting pretty good. Where you see these like yellower colors here, we are seeing relatively good, almost up to a sort of doubling of the nitrate load reduction at this sort of flow rate temperature combination. This is what it looks like the reactor is doing, right? The gray areas show where the reactor is drained. It'll flood, the rates will decrease, it'll drain, rates will increase, and then you'll see this pattern repeating. You'll see it here as well. One thing you'll note is that at the higher flow rate scenario, there's fewer drainage events than there are in the slower flow rate scenario, and that's partially because when the flow is high, the cost of draining is higher, right? Because you're missing more of that nitrogen load for every unit of time that the reactor is drained, okay? And so what's happening over here? Why do we see these zeros? What's up with that? So this is basically telling us that in this region, the reactor is already getting 100% removal without a need for draining, because these are high temperature, low flow periods where the reactor would be working really well just at baseline. So there's no need to do anything else if you're in this region. Okay, so what we could then do is we could also run this in a non-steady state mode, and we could directly compare a simulation using the Markov decision process to govern the draining, reflooding cycles, and compare that to the data that we captured when we did our exogenous carbon dosing. Okay, so this is the Markov decision process model, driven by field data from the spring of 2023. And so the green line, this is showing us our baseline model, what's happening with no control. The orange is showing the rates that we achieved with exogenous carbon dosing, and the black is showing our simulation results using the wetting drying cycle. So what we can see from this is that we do get higher rates from the exogenous carbon dosing. The exogenous carbon dosing works better, but also at much higher cost and with more environmental risk in terms of released acetate, whereas this wetting drying system, all you really need is a valve at the outlet to open and close to drain the reactor. Much lower cost, much lower risk of, fewer chemical inputs, less need to add things to the reactor that could then leave the reactor, possibly biofoul the reactor if there's too much biological growth. Okay, so we can see that the drying reflooding got us a cumulative load reduction that's somewhere in between the baseline scenario and what we saw with the exogenous carbon dosing. And so basically, we can see that exogenous carbon dosing meaningfully improves nitrate removal rates in challenging times, but there are risks, and it's expensive. Okay, the deliberate draining reflooding does provide an alternative control method. It's not as effective, but it's lower cost. But one thing I forgot to mention is that in order to actually optimize this, it requires knowledge of future discharge scenarios. Okay, when we ran this with our non-steady state version, we used like a 2-week decision time horizon. So we assumed that we knew what the discharge and temperature would be 2 weeks into the future and made decisions based on that. And obviously in the real world, that would be imperfect information. Okay, so we might not be able to implement this optimally in practice. Okay, so anyway, now we have a new project from USDA. I'm super excited. That's why Jihao and I are here. We're going to build on some of this work that we've done on real-time control, implement this in Blue Earth, Minnesota. This is the largest woodchip bioreactor in the United States, possibly in the world. So I'm really excited to take some of these ideas, test them out here, and also hopefully achieve some economies of scale, right, to be able to implement this at larger scale. And this is a great collaboration with Gary Feiereisen, who's with the USDA ARS in St. Paul. Okay, so time is, okay, so anyway, challenge 1, I said hydrological variability leads to inconsistent treatment performance. Okay, maybe we can dose carbon. That can help with this problem. Challenge 2, breakdown of woody biomass is slow in anaerobic conditions. Okay, just drain the reactor, expose the wood chips to oxygen. This helps us with this challenge. But there's this third challenge, which is not a technical challenge, which I didn't talk about, which is the challenge of uptake by farmers. Okay, because farmers have very little to no incentive to actually install these things in their fields. And this brings me to the third part, which will be short, about rice paddies as treatment wetlands in a circular nitrogen economy. Okay, so this is really new work. Normally I wouldn't talk about this in a seminar like this, but I wanted to do my best to add some elements of circularity. So you're getting a little sneak peek here on some things that are in their early stages. And the way this really emerged was that farmers in central New York around Ithaca came to Cornell Cooperative Extension with concerns about flooding in their fields. They were losing crops to floods. Flooding is happening more frequently, it's more intense. And they were starting to take a lot of their land out of production, okay, because of heightened flood risk. So they approached Cornell Cooperative Extension about rice as a flood-tolerant crop, okay? The first few folks who worked with this were some colleagues over in the Ag College at Cornell. And the cool thing about this is this really is kind of like a bottom-up effort, coming from local farmers. with this idea of growing rice. They sort of then contacted me because as another part of my research program, I do some work on arsenic in rice paddies. You may or may not be aware that arsenic uptake into rice is a challenge for rice agriculture. Accumulation of arsenic in rice as a food product is a problem, especially for kids. A lot of children's infant crackers are made with rice flour, so FDA has specific action on this. But because I had some experience doing field sampling in Arkansas, measuring arsenic in pore water, so I sort of got involved because of this. Methane emission from rice is also a major sustainability concern. And so basically now let's arsenic 101. Why is there arsenic in rice paddy soils? Okay, this is an XRF image. This shows like a soil mineral. Green is iron, red is arsenic. Yellow is like the iron arsenic overlap. You'll see a lot of yellow on the surface of some of these mineral grains. That's because iron is a good adsorbent for arsenic, but when we get into reducing conditions, the iron releases the arsenic, can get reduced to a more mobile form of arsenic-3. And sure enough, when we sample pore water from some of these experimental rice paddies in New York, we see this relationship between iron and arsenic, dissolved iron, dissolved arsenic in the pore water. Okay, there's not a direct link between like the drinking water limit and the pore water concentration. We're not talking about drinking pore water, but this does provide an indication of, okay, like what's like an elevated arsenic concentration. So you can see we're seeing high concentrations of arsenic in the pore water. We haven't yet analyzed the concentrations in the grain, but based on my experience, I'm expecting if we see this much arsenic in the pore water, we're probably going to see some relatively high concentrations in the grains. What does this have to do with nitrate? Okay. Redox ladder. Okay, we have nitrate higher on that redox ladder. So basically, we can add nitrate to the system. We can oxidize iron. Okay, basically reverse that iron reductive dissolution reaction. The arsenic can sorb onto that iron surface. And basically, if we can maintain a redox potential poised in this direction, we'll inhibit iron reduction and also, importantly, decrease methanogenesis. This has been shown before. It works. So the idea here that is really in its early stages was really born out of the fact that there are these pilot rice paddies here very close to where I've been working for a couple years on our field scale woodchip bioreactor. So the idea is, can we just divert that tile drainage to the rice paddies? Think about the rice paddies as a form of treatment wetland, okay? It's a carbon-rich anoxic environment. Denitrification will be quick. Maybe we'll even be able to offset some of the synthetic fertilizer needs that are used for these fields. My colleague Chuan did a geospatial analysis to think about, okay, it's cool, we can do this on our little experimental farm. What's the broader potential for this, okay? So they did an analysis of flood-prone agricultural land. It's a little bit hard to see here, but one thing that was kind of interesting about this is it really picked out the eastern Arkansas rice region, which is where most rice in the US is grown. It really did pick that out as like a suitable area for growing rice in sort of flood-prone agricultural areas. But we also saw high areas of suitability in sort of like the Corn Belt here, which suggests that there could be potentials to implement this in areas of the upper Midwest. And this up here, I saw this and I was like, okay, I got to talk about this in my talk because none of us have any idea what's going on with this situation up here. But hopefully someone here can enlighten me as to like why there's this like hot spot for suitable land for growing rice at the Minnesota-Dakota border. So some of the main points here, and this is why I'm pretty excited about this because like as someone who does like redox biogeochemistry, I'm like, okay, we can use nitrate. It's a waste source of nitrogen. We can diminish arsenic uptake by oxidizing iron. Maybe we can also suppress methane emissions by keeping that redox potential high. And this would be a great reuse of some of that nitrate-contaminated water. Huge research needs here. Okay, what's happening of the N that's going into that soil? Is it being taken up by plants? Is it denitrifying? Is it producing N2O, right? This is a risk if we're dumping a lot of nitrogen into this field. And stay tuned. We just got some funding from NSF to keep doing this, so that's exciting. And I'm excited. For more information, check out those papers. Thanks for your time. I think it's about 5 minutes for questions. Thank you.
We have time for several questions.
Thank you very much. That was a beautiful piece combining engineering and science. I am, however, a little disappointed by your title. You said non-point source, but everything essentially was coming out of one pipe. My experience with tile drains, a farmer has 160 acres, you're going to have lots of spots where it's coming off, and you'd have to replicate that at each of those. Have you given some thought to how to actually implement this in the real world?
Sure. So yeah, does a tile drained field represent a point source or a non-point source? That's a good point, because essentially when we have tile drain systems, we're taking drainage from an area of maybe 10. This site in Blue Earth is 200 hectares, and it is routing it into a single outfall. So I appreciate your point. Maybe it would be more appropriate to talk about nitrogen in tile drains rather than truly non-point. But I still think that when we think about agricultural nutrient release, I think generally most people would think of this as especially from row crops. I think most people would still think of this as a non-point source, even though there is a single outfall. And that's the beauty of it, because that allows us to do something rather than if it's truly non-point, it's really hard, and you can think about riparian buffers, you can think about things like that, but there is an opportunity created by this sort of let's call it a hybrid, intermediate point source. But I appreciate that point.
Yeah, thank you for your nice talk. You mentioned about carbon dosing and wetting and drying. But I think there are other potential options, such as inoculating microbes that do that job for nitrate removal or carbon degradation. And another potential approach is to aerate the bioreactor. Do you have any opinion on those?
Can you say that again about the
inoculation of the microbes and aeration of bioreactor?
So inoculation, so bio augmentation? Yeah, yes. So I don't think I have the slide here as a extra slide, but in the paper that we published in Environmental Microbiology, we did construct some metagenome assembled genomes, and we were able to look at I think about 100 mags that we assembled from the reactor microbiome, and we sort of classified them as to are these denitrifiers that can degrade cellulose and hemicellulose? Are they cellulose degraders that don't denitrify? And we compared the woodchip community to a community from activated sludge wastewater treatment plant, just as having some comparison. What we saw, which was interesting, was that most of the denitrifiers in the woodchip bioreactor already had cellulose degrading capability. Okay, so in that sense, the environment I think had already selected for a community that had that capability. And when you look at the activated sludge environment, there was very little capability for lignocellulose degradation there because they don't need to do it. So I guess I do tend to believe in everything is everywhere and the environment selects. I think that's sort of my approach. So that's my thought about bioaugmentation, but
Yeah, we can increase
yes. Yeah. Totally. Definitely. What's the persistence of that? How long is that going to last? These are important research questions. But of course, I think it's also an all hands on deck approach. We need to improve things. And to the extent that this can be done at relatively low cost, I think it also, and with low risk, I think it also should be done as part of the menu. Thanks for the question.
Hi, Matt. Great talk. Really enjoyed it. You showed N2O concentrations, but you didn't really talk about them. And it looked like your Nazi, I don't know if it was numbers of genes or expression of genes, but those that was consistently high,
Yeah.
but yet you saw this spike in N2O. And
Yeah.
I wondered what was going on, because it should, during that period, my understanding was it should stay anaerobic. And so what's going on with N2O? And
Yeah.
obviously you're measuring it, so
Yeah, yeah.
what's happening more broadly with that?
Yeah, thanks so much for the question. In that paper, in Environmental Science, Water Research, and Technology, we did also measure N2O, and we saw that the wetting drying led to lower N2O, dissolved N2O, leaving in the effluent. And the understanding there was sort of like more carbon were driving the denitrification further to completion. You're right that we saw NOZ expression low at that first time point at the 0 hour when there was the highest oxygen. NOZ expression was low, and then it bounced back and was pretty high for the period after that. So, and there was this transient increase in the N2O product yield. So why is that? Was that sort of like a lagged effect of this sort of like suppression of NOZ expression very early on when there was oxygen? Could there have been some sort of like post-transcriptional issues? I'm not like truly like a nitrogen sort of cycle microbiologist. Satoshi probably has better insights into that than I do. But those are some possible explanations. But I think the key point was that we actually did see a decrease in N2O product yields from the wetting drying cycle, when initially our hypothesis was that, okay, we're adding oxygen to this denitrifying environment, we're getting maybe some anoxic interfaces. We expected the N2O to actually get higher, and we saw the opposite. So we saw that as like another benefit of this approach. Thank you for the question.
Yeah, great talk. I appreciate it too. The wood decay is a pretty slow process. The chips, the crystallinity index looks like it's pretty sort of around the edges
Yeah.
on those that are losing it, and also the reaction of the reactor seems pretty fast in terms of lags and stuff. Kind of not in line with what I would think for wood decay. So it seems like maybe kind of a superficial dynamic on the wood chip. Is that true? And could that, in terms of surface area to volume, the size of the chips that go in there, could that? I'm sure that has some benefits and cons, but is that true? Superficial dynamic there, or
So do you mean
surface dynamic, I guess, on the chips?
are you sort of asking like, is that wood decay fast enough to actually support the denitrification rates that we're seeing?
No, is most of that activity that is supported, or the labile carbon, is that pretty much coming from that outer edge of the wood chips, and is most of the internal part kind of just dead to the world? Is it
Totally.
wet? And
Yeah, that's our understanding at this stage. I mean, this is also relatively new data that we haven't published yet, and we're still working on. But that's sort of what we're taking from that, is that it is very much like a surface-driven process, which is not. It's not surprising that this thing would sort of like degrade from the outside.
Right.
And it seems like, yeah, that interior core, maybe around the pores, there's a little bit of action there. The interior core that doesn't seem to have that much degradation. In another lab group at Cornell, a student just did a paper where she did like a biofilm extraction of a wood chip, and then she ground up a wood chip and did metagenomics with sort of like the biofilm extraction and then the ground up wood chip. And she did see quite different communities, like on the surface versus the interior. So that also kind of supported the idea that there are really kind of like different processes occurring at the surface versus in the interior.
Okay, maybe one last question, and then we have an online question.
I actually had two questions, but I'll shoot the first one. So that is, can you actually add different other types of lignocellulosic biomass that probably has a lower, crystallinity? Like cellulose comes in different amorphous forms too.
Yep.
Would you do that to manipulate the kinetics of your reactor, and like how wise would that be? So that's my first question.
Okay, let's start that one. Yeah, totally. You can use that. There's a lot of interest in like corn cobs. Okay, corn cobs is another like waste residue that's really very abundant, especially on these fields. Traditionally, the thought has been that like wood's good because it actually decomposes kind of slowly, so that the bed lasts a long time, and you also don't end up leaching too much carbon. Whereas like other carbon sources with a lower like lignin content will degrade like too fast, possibly. But there's some really new results coming out with like corn cobs that are showing corn cobs also lasting for a long time and supporting higher rates for a long period of time. There's another approach, which is like you put something really like degradable upstream, okay, and like maybe like a removable cartridge, that degrades faster, releases DOC, which then moves down the bed and then can provide carbon for some of the downstream biofilms. So there's a bunch of different ways that you can think about using other types of lignocellulosic wastes, integrating it into the system. But that's a great question, and I'm super interested actually in using this X-ray scattering technique on corn cobs, actually, and seeing how like something like a corn cob is degrading separately than something like a wood chip. So maybe in a year or so I'll be able to talk about that.
One last question from online audience. They are interested in whether there are some varieties of rice be more suited for these paddies, and are there rice breeding to optimize this system?
Yeah, great. Yeah, thanks for that great question. So one of the people who was first approached about this project is someone named Susan McCooch, who's a plant breeder at Cornell, really famous, like National Academy member. So she was like all over this, and she was like, I know about the varieties in like Japan, northern Italy, that do grow in like temperate environments. And what we've seen is that the rice is growing well. They're getting a good harvest. Last week we had like a sushi taste test with some of the rice that we grew there compared to rice like from the restaurant. And at least for me, I couldn't tell a difference from which one was which. So yeah, it is growing, but yeah, there's a lot of work that needs to be done there to like really kind of scale this up. All the system changes that need to happen, farm machinery, et cetera. So that's kind of outside my area. From a soil biogeochemistry perspective, I think it's super, super cool. And also, we'll see where we are in a couple years with that.
Thank you so much. Let's thank our speaker again for the great talk. And thank you all for coming.